Да, Игоря Леонидовича Чернявского, который работает в Манчестере, в университете Манчестера, на факультете математики и одновременно, как здесь указано, сотрудничает, значит, с медицинским центром. И Игорь расскажет нам a story of two circulations, and it will be in English. So, я попросил Игоря, чтобы он рассказывал не торопясь, чтобы и научная сторона с точки зрения языка, чтобы это было понятно нашим слушателям. При необходимости мы можем делать такие небольшие паузы и давать какие-то пояснения или задавать вопросы. Поэтому, пожалуйста, Игорь, мы вас с интересом слушаем.
Thank you very much. So I would like, well, good afternoon, everyone. I'm very happy to be here, and I would like to thank Vitaly Isaacovich and Anastasiu for present this report. As explained, I'm presenting in English, and the reason is that materials and the terminology I'm using suggest that my view.
Я прошу прощения, что-то какие-то проблемы со звуком. Это только у меня или... Анастасия, вы хорошо слышите?
Нет, тоже пропадает.
Тоже.
Игорь, пропадает все время звук. Старайтесь говорить, пожалуйста, может быть, ближе в микрофон.
Can you hear me now better or not?
Yeah, it's better, it's better.
So what I'm saying is, I would like to thank Vitaly Isaacovich and Anastasia for inviting me to this seminar. I'm very happy to be here. And I'm presenting this in English because all the materials I have are in English. But I'm more than happy to answer any questions if you would like to ask them in Russian. And again, as Vitaly Isaacovich said, please feel free to interrupt me at any time. Is the sound better or it's still interrupting?
Ну, сейчас хорошо.
Okay, thank you. So with your permission, I will continue. And today I would like to tell you about a unique system, the human placenta, and in particular, the flow and transport phenomena, processes in this organ, which is absolutely unique. And I hope by the end of this talk, you'll share my enthusiasm about modeling such a system. So before I start, I would like to acknowledge all the many collaborators I work with on theoretical side, experimental side, and clinical side. A majority of the results I will show are done by the people highlighted in red, by Alexander Ehrlich, Philip Pierce, and Win Toon, who worked with me over the years. And this supported by the funders, by the Medical Research Council in the majority of the results I show. I don't know how many of you read this book by Erwin Schrödinger called What is Life? The Physical Aspects of the Living Cell. So this is a very nice book, and it actually shows that Schrödinger not only a founder and father of the quantum mechanics, but also he had a very deep insights into modeling biological phenomena. And indeed, by just using some of the ideas from statistical physics, he anticipated in this book, the existence of the DNA before it was experimentally found as some sort of aperiodic crystal. But also, he was arguing about the role of physics in modeling life and other natural phenomena. And for example, he saw chemistry as the physics of small scales and astronomy as a physics of large cosmic scales. And I would like to argue that the biology is a fundamentally physics of the multiple scales which are interlinked and cannot be seen as a dominantly one skill or the other, but rather as their combination. And you probably saw that picture by Da Vinci, where he was studying the development of fetus in the womb. And it is actually very anatomically correct, with lots of details, still recognized as modern anatomy. And it was 16th century. But later on, some facts being mixed up less correctly. For example, with the invention of microscopy, people started to look into details of images such as the sperm, and there was a concept of homunculus, or little human, where people thought that there is literally little humans are already formed and just growing, attached to the placenta to nourish them and help them grow. And this brings us to our modern times, fast forward to the 21st century, where now people started to develop all over the world the artificial placentas, where they can support very premature fetuses outside the womb. See on that photograph a biobag recently published by one of the American groups, similar research done in Australia and Canada.
Hello, Mr. Igor.
Yeah?
Please, I can't hear you. Can you improve your sound quality?
Give me one moment.
Okay, thanks.
I think that if you speak in the microphone, it is okay. So you should be really close to the microphone.
It normally works all right with Zoom. Maybe there's something with Microsoft Teams, which is special.
No, now it's very good.
Yeah, now it's good. I don't know what is happening.
Yeah, I don't know today. Give me a moment. I just want to check the settings.
Or maybe when presenting, you lie on your back.
Can you hear me all right now?
Yeah, it's better.
Is this still all right?
Yes, it's okay.
Better than before.
It's a bit strange. Okay, I'll try to really speak closer to the microphone and see if it will work.
Thanks.
So please do tell me if at any point sound drops.
No problem. I will be your watch.
Yeah, thank you. Appreciate that.
Welcome.
Okay. Right. So what I was saying, that we start with Leonardo, then we move to the other researcher who looked into microscope and thought that inside the sperm there are little humans. And now, in our modern days, we of course understand much more about development, and there is now even some attempts to effectively recreate conditions outside the womb to maintain development for fetuses which don't have lung mature enough so they cannot breathe on their own. So that's artificial placenta is already reality. It's no longer science fiction. But all of these three pictures, they're united by the concept that they all supported by external environment, which provides them oxygen and gases and other essential life support. And that's all provided by placenta. So to really better design artificial placenta, we need to understand what real placenta is doing. The placenta is absolutely unique, I have to say, among all mammals. So for example, mice and humans have somewhat similar lungs or heart, but their placenta is totally different. So there is no real animal analog to the human placenta, which is important to bear in mind. And that's why modeling approaches and laboratory experiments are very important. But on the other hand, human placenta is readily available for experiments. It's very easy to get hold of the placenta. So you can get it into the lab after delivery because it's a clinical waste. So you cannot easily study human lung, but you can easily study the placenta. And of course, it's a critical link between the mom and her baby. So if anything goes wrong with the placenta, then the fetus has no chance to survive, to develop health. So in brief, it's an interface composed of complex structures, highly convoluted trees, which are interfaced with the maternal blood, containing nutrients like glucose, amino acids, and so on, and of course,
Hello.
hello.
The sound. Okay, now is I don't know. Oh, is it only me? I can hear you.
Can you hear me now?
Yeah.
Is it just dropping sound or some noise?
Yeah, sort of. Like if you are speaking, then it will be the sound quality will be reducing to nothing.
Okay, I think it's something about Microsoft Teams.
I've never experienced this before. I've been using Microsoft Teams for more than a year now.
Okay. Well, I'm speaking to the microphone more or less. So it's very interesting. Why is it doing that? I'll just double check if there are any settings and I can check in the Teams. How does it sound now? Is it okay?
На коротких фразах хорошо.
Right.
Can it depend on the language you use? Maybe if you speak Russian, the sound will be better.
I don't think so. Maybe you really want Russian.
Yeah, so the Microsoft Teams doesn't support English, apparently.
The funniest thing I've heard. So.
Yeah, please continue. We hear not okay now.
Okay, to me it shows that it's working, but maybe it's something happening indeed. Let me just. I don't know if in any settings you can advise.
No, I don't think it's related to settings. Maybe it's either connection or microphone or something like that.
На самом деле, если у вас внешний микрофон и в компьютере микрофон, они могут конфликтовать. И там есть выбор между двумя источниками.
Yeah, I choose the external one. Yes, this one.
Yeah.
Yeah. So it's might be some noise suppression happening.
Да, тоже может быть. Да, лучше отключить, наверное, его.
Okay, I canceled noise suppression. Maybe it will help. Let's see. Thanks a lot. Apologies for that. Sorry, I'll go forward. So the structure. Can you hear me all right so far?
Very good.
Okay.
Is it okay? Is the sound okay so far?
Yeah, it's okay. It's okay. Please continue.
Thank you. Sorry. So what I would like to show you is a short movie about the different scales of the human placenta. So you'll see moving from the scale of almost a millimeter to a micron. So that's the data acquired at the synchrotron, which is the powerful X-ray tomography. So you can see if I pose it the cube of tissue, and you can appreciate the complexity of that tissue, composed of maternal and fetal components of the placenta. And if we start zooming in a minute, looking at the smaller region, we can try and segment this into different domains. So first, again, if I'll pause, if I'll pause here, you can see the porous side of the placenta, which is itself very complex porous medium. And then we will proceed to the fetal side, which will be in purple. So you see the network embedded, which is a dual domain. So there's at least two key features, the maternal and fetal side. So you can see it in blue. Again, if I pose it. And that is a network, which is the fetal side of the human placenta. And then we can zoom in further and see what is the functional interface between these two networks, where all the interaction between the maternal and fetal circulation occur. So you can see that this is a scale of about 100 microns, which is the width of the human hair, approximately. And that is the capillary level, where at outside of this is the maternal blood, and inside these blue domain, blue vessels, is the fetal blood. So you can appreciate the many scales and the complexity of the structure. Now, that's just the same idea, but expressed in a summary form. So you can see on the left the whole fetal vascular tree of the placenta, from the umbilical cord at the bottom, bifurcating into fine, and more fine branches. If you take a cross section and take corresponding tissue, you'll get this porous medium. So these green are the vessels, cross sections, and outside is the maternal blood. And if you'll zoom in, you'll see that exchange unit, which is interface between the mother and her baby. And it goes from the scales of centimeters to microns. So it's 4 orders of magnitude in spatial scales. And today I'll focus on both aspects of the network side, the fetal side, and the maternal porous side. So there'll be 2 stories to the placenta, the maternal flow and the fetal flow. So first, let's talk briefly about the function. And on the function, I mean the solute exchange and transport. Here you can see yet another picture. This time it's electron microscopy, where you can see the actual interface, where that's the red blood cell erythrocyte from the fetus, from the fetal side, and outside will be maternal blood, but you don't see it. And that's the thin boundary separating the two circulations. So there is some analogy between the lung, but in the lung there is an air-blood interface. Here there's a blood-blood interface. And of course, as happens, then you deal with the biological structures, you have all kind of energy-dependent transporters, but also more simple transporters for simple solutes, such as oxygen and water. So I'm not going to focus on the complex transport, such as amino acids or immune complexes, but for simple solutes, such as water, oxygen, glucose, the approaches we developed are widely applicable.
Can I ask a short question, please?
Please.
Erythrocytes, are they also exchanged or they're separated between mother and the baby?
It's a very good question. They are not exchanged unless in extreme pathology. So the coupling only through the solutes. The red blood cells do not cross the barrier.
Okay, so all blood cells, in fact, are produced in the fetus, right? I mean,
Yes, that's right.
okay, thanks.
And always then we are starting to analyze a new system. It's helpful to think of analogies, which are perhaps better studied. And one such analogy would be a blood-brain barrier, because again, you have two sides. One is the general body circulation, another is a brain circulation, and the body tends to protect the brain. So there is quite a tight barrier separating the brain circulation and the rest of the systemic circulation. And for many pathologies, for drug development, it's important to understand how to cross that barrier, how to get drugs delivered to the brain, or in another case, how to protect brain from toxicity. And what often done in these situations, for example, by a pharmacy, pharmacology industry, they tend to treat their drugs as two-dimensional space. One is permeability, so how easy you're crossing this barrier, this cell, say endothelial cell, and second, how easy is the particular solute is dissolved in this kind of membrane. So it's very much proportional, so in log-log scale, to the relative solubility of the compound in oil or water. So because membrane is effectively oil, so the more it's soluble in oil, the easier it will be cross this membrane. And some of the solutes are more neutral. But sometimes you have some outliers which are crossing much more than you expect, and some of them cross much less than you expect. And the reason for that is because you have either transporters which provide you energy, so for very large molecules which wouldn't cross easily themselves, to facilitate transport. And that's the case for glucose. Another case, if it's a toxicity, and there are often defense mechanisms in the cells, in the body, to actively pump out undesirable compounds, undesirable solutes, so that there are kind of a negative pumping. So it tries not to let things cross actively. And that's why you have things lying both below and above, kind of this generic trend. And the aim is ultimately to try to develop a similar understanding of how different solutes, both kind of normal, produced in the body, and chemical, which relates to the drugs, which are administered to the mother, cross the placenta. And I'll say, very briefly mention a few points about development of modeling. So it started quite some time ago, but it was treated as a black box, mostly motivated by the complex chemical interaction, but ignoring all the spatial information about how the solutes are distributed within the organ. So they treated it as a well-mixed black box. Then, relatively recently, I say, maybe about 5-10 years ago, there was a series of models which became spatially aware. So they started to understand the structure of the flow and transport, but in a very idealized setting. Thinking of placenta as composed of regular cylinders, or just as a porous medium, uniform porous medium. And then, relatively recently, in less than 5 years, and literally in a very, very late 15th afterwards, there is an explosion of image-based modeling, partially enabled by rich imaging data coming from all modalities, from X-ray tomography, from microscopy, and many others. And you can see that all features of placenta can be captured, but they are intrinsically very variable, very complex, and hard to characterize at scale. So we can capture a small portion of a large organ for one patient and model it, but it's really not producing enough statistical variability to really appreciate how robust are predictions based on this one sample. So what the aim of the approach we undertake is to try to revisit the available information from images and learn what are the key features which we need to collect and preserve, and then return back to as simplified, upscaled models. So we effectively want to revisit this black box, but with the information we learn from detailed microstructural features. So we want to know how microstructure affects the function. So the first story is about the maternal transport and flow. So you've seen the placenta. It's very complex, and it's often helpful to think, can we make a simplest possible model for the maternal side? Well, if you take a cross-section, in green you see the blood vessels of the fetus. Outside the black space is a maternal space, and we want to treat it as a porous medium. And often then you see it as a porous medium, you try to identify some effective properties, such as permeability or diffusivity, and extract that and then work at the larger scale, at the tissue scale. Because we have images, there's established a technique of asymptotic homogenization or averaging, is it known, or upscaling, which allows to go from detailed physics at the macro scale to the simplified description at the larger scales. So here we go from the Stokes flow to the Darcy flow, and information which we obtain to connect the two is the permeability tensor, which characterizes how easy it is to flow in different direction. And that's entirely defined by the geometry in appropriately scaled formulation. But that's just the mean description. Of course, there will be also the fluctuations about that mean. But if for a moment we only think about that we found what is the effective resistance to flow of the human placental tissue, what can we do? Well, we can formulate the organ scale or tissue scale model, but saying, okay, if I look at the maternal vessel, which you can see here, entering the porous space, which you appreciate quite dense, it delivers a flow and solutes, and then it will be drained by the veins. So that's the maternal side, that is the fetal side on top, and placenta connects them. Now we can think that that is a unit of exchange. So one artery and two veins, and around them is the dense tissue where blood percolates. And if we think of the characteristic numbers, it's a very low, relatively. Reynolds number is probably maximum we can get. It's actually likely to be less than that, far away from the artery. And the Peckley number is quite high, so it is dominated by advection, not by diffusion. So Peckley number, just to remind you, it compares the strength of the flow to the strength of diffusion. And Reynolds number shows the relative importance of the inertia. Now, if we think of that unit of exchange, again, it's as simplified as a possible way. So we have a source, let's say point source of flow and solute at the base, and two sinks, the veins, which drain that area, that domain, and we assume no permeability over this shell, the hemispherical boundary. So then it's described by two parameters, the Dunkel number, which compares the relative strength of the uptake, so how strongly it's absorbed, in a sense, by the fetal tissue inside, and also we can also vary the distance between the artery and the vein at the maternal side. Right? Well, that's all overly simplified, and it's as simple as it can get. So in a sense, we started with extremely an unmanageably complex system and reduced it to a hemispherical domain with the point sources and sinks. So it does sound like a bit of a spherical cow or horse. But I will show you that even the most simple model can be still insightful. And the benefit of it would we can compute the flow field directly, exactly analytically, and also learn what is the corresponding concentration of a given solute. Let's say oxygen. So the blue here are the streamlines. So you enter from this source and then you go back to the vein. And the green are the isobars, the lines of constant pressure. Again, no permeability assumed at the upper boundary. And that's the solute. So you enter with the maximum concentration, the maternal arterial blood concentration, then you leave through the veins. And that's what we have.
Okay, excuse me. Here you consider two-dimensional problem?
No, it's three-dimensional, but it's axisymmetric. So we consider it a spherical, hemispherical domain. So we assume that it has axial symmetry. So it's symmetric with respect to the. So we have one source, two things along the line of symmetry.
Yeah, but the things are pointwise. They are not radially symmetric. No, the things. It's not a circle. Sync is not a circle, but it's a point, right?
Yeah, that's a point. There are point sinks on the line of symmetry. Yeah, and so in a sense, that is
so
just
there is no radial symmetry in this problem.
There is axial symmetry, no radial symmetry. Yes.
Yeah.
So that is a cross section. So we can move it off plane, and the picture will be the same. So that's a cross section through a hemisphere. It's an idealization, of course.
Yeah, thanks. Please continue.
And just to illustrate that even that highly idealized model is still useful, we use it to explain observations in experiments known as the smoke rings, where it was done in monkeys and even in humans, unfortunately, because people didn't know about danger of X-rays in pregnancy. That was done in no ethical committee ever approves this nowadays. So they injected a dye, which is visible in X-ray, in X-ray movie, and they captured how it appears. So they injected it upstream and saw how it emerges in placenta and then propagates and disappears. So they saw as a time lapse captured pictures how you get that rings forming and then expanding and disappearing. And there was speculating about what does it tell about the structure of placenta and so on. And there was idea that it's a cylindrical empty space inside these units in placenta, which are called placentomes. And in fact, if we just assume a uniform porous medium and introduce a bolus and a shell of dye, which you can see with the pictured with the blue lines, the dotted blue lines on the panel A, and then you project X-ray from the top, then the intensity will be proportional to the relative thickness, which is shown here in red, because you shine X-ray from the top, the highest thickness will be close to the periphery, and then there will be less thick at the center. And as it expands, so you can see, so these points are the source and sink, so that's the artery and there's two veins, so point sink. So looking from the top, and you can see that it forms a ring indeed, which propagates towards the veins. And you can measure the area of that ring as a function of time. And that roughly matches what they observed experimentally. And we didn't need to assume any heterogeneity or special structure inside the placenta just by understanding what the expanding shell of the dye will do if you look at this in X-ray in the simplest model of porous medium with the flow supplied by the point sources and drained by point sinks.
Excuse me, the assumption I don't understand. At the top figure, you have this red curve. Is it the thickness of this die?
Yes, it's a thickness of the die projected on the base. So if we look at the vertical projection, yes.
Yeah, but why that it is larger from the sides than in the center where you have the source?
Because we produce we don't introduce a constant supply. We produce a finite volume of the die, and then it expands. So at the beginning, it is introduced as a ball, but with time, that front will propagate into the. So it follows isobars, in effect, of the flow. So
yeah, but why the form is like that? This is what I don't understand yet. Why it is not convex, for example?
Well, because the thickness we measure is the thickness between the front and the rear of this die. So that is the thickness we measure. We measure thickness between the front of the die and the rear. So we conserve the volume of the die, and therefore it becomes a thinner and larger ring.
Yeah.
And the thickness in the middle, just above the source, will always be smaller than the thickness if we look at the cross section.
Okay, so the red line measures the difference, right?
Yes, that's right.
Okay, thanks.
It's not the distance to the base, but assuming the finite volume of the die, what is the relative thickness at the center compared to periphery, looking from above.
Okay, thanks.
So in fact, the term smoke ring is incorrect because there's no vortices, so there's no rotations of the fluid. It's just literally tracing the relative thickness of the die in X-ray, orthogonal projection, projecting on the plane, on the base. So that's just to show that this model makes sense, and it can capture qualitatively and to some extent quantitatively what they observe in terms of how long it takes for dye to reach the veins. And other way we can do is we can think of what is about uptake, what is the measure of uptake efficiency. So we can compare the relative contribution of density of tissue to the total uptake of solute per unit time. So the total uptake rate, which is the arterial concentration flux minus, so what came in minus what came out of the maternal side of placenta. And in fact, it's not totally surprising that there is an optimum density of the tissue, because for very high density, there will be very high resistance to the flow. So the flow will be very slow, and the net uptake rate will be small. So it will be very hard to deliver material into the placenta. On the other hand, if the density of placenta is very low, the tissue density, then there is a very high flow through the maternal side, but also there is not enough surface area of the placental interface available to extract that solute. So again, we will have a relatively low amount of uptake. So there is an optimum tissue density in between, and it's true for a variety of reactions. So we can assume that reaction is linear or nonlinear at the interface, but the principle remains that the volume fraction of the tissue relative to the total volume of the placenta does have a significant impact on the net uptake. So with this, I would like now to start thinking about, okay, we learned a bit about the average properties and simplest upscaled model, but we still don't connect it much about detailed features of the microstructure, only perhaps through the densities or volume fraction. And since we have very rich images, the question is, first of all, how would you choose the right scale for extracting these features? And then what features would we like to look at? So here you can see a cross-section, stained cross-section of placenta, where the blue is the tissue and white again is the maternal space. And we can change the size of this window and see how the volume fraction varies. So that fluctuates. Of course, if you take very small window, it will be pretty much a random quantity, but then it converges to something more deterministic, more homogeneous at the larger scale. So there is a generic principle that we should or expect to observe some macroscopic homogeneity beyond given scale, unless there is some macroscopic heterogeneity of the organ, which is also possible. And we can do it a bit more quantitatively, more carefully, by utilizing these rich three-dimensional data which we just extracted in synchrotron. And you can see, for example, as we move from slice to slice, just looking at the area fraction, so relative fraction of tissue to the maternal space, that it varies quite wildly. But as you increase the region of interest to extract the features, such as volume fraction, the error diminishes, so it decreases, so the fluctuations becomes less significant. But the question, how do we define what is the reasonable size of that region of interest? Because we cannot go to very large sizes, it's not feasible. And also we want to understand at what point transition from microscopic variability to mesoscopic tissue scale and then to macroscopic organ scale occur. So what are the scale separation in the system, if there is any? And at the bottom panel, C and D, you can see again that the relative variability, the fluctuations, they decrease. And in fact, we can show that for sufficiently large volumes, they decrease inversely proportional to the square root of the volume, both for the volume fraction and say for the specific surface area of the interface. And on the right, on the panel D, you see the so-called correlation function, two-point correlation function, which tries to see how much there is interdependence between the different points of your medium. And we again can see that it disappears at around 200 to 300 microns, which is consistent with what we saw previously, about 1 millimeter, even less. And that is quite useful. So now we have a more robust way to quantify at what point transitions from microscopic noise in terms of structure to the more mesoscopic and macroscopic tissue level. And it's also helpful to think of another idealization. If you don't want to keep the entire shape, but only preserve the center of mass, from each of these tissue pores, we can think of conceptual different arrangements. Again, think of 2D for simplicity. We can think that there's absolutely no structure, absolutely random, uniformly random or Poisson process. Or we can think other extreme as absolutely periodic, regular structure, is often assumed in averaging, in homogenization. Or we can think somewhere in between, where we have a hardcore, where you take a random structure, but you do not allow for the pores to come too close to each other. So there's some minimum distance, constrained by the structure, by biology. Or you can say that there is some underlying structure, but it's disturbed. For example, you have a periodic square array, and you displace randomly according to the normal perturbations each of these points. And there is a way to quantify the randomness or clustering in these structures by using a function related to the two-point correlation, an integral of it called K function, which effectively saying that if you have a totally random medium like a forest, which is where this came from, then you expect that that function will be proportional to the area. So it's exactly number of points you expect from a given point arbitrarily, and that will be just proportional to the area, and the square root will be linearly varying with the distance. Okay, so that's what it measures for absolutely random medium with no structure. For a hardcore, where the minimum distance you don't allow to get closer than, you'll get that threshold immediately. You can see it, and if you compute it, and then it converges to the uniformly random for larger scales. And for normally perturbed, because you have intrinsic scale, length scale of the array, so that kind of wavelength given by the period of the array, even if you disturb it, you get this oscillating structure, which practically repeats itself at the scale of the array. Now, if we look at the placenta, again, take that section and take a center of mass for each of these little blobs and compute what is the integral of this two-point correlation function, we see that it's indeed somewhat in between hardcore and uniformly random. So it does has a threshold, which then transitions to the uniformly random, and again, transition is comparable to what we've seen most recently in 3D, which is about 100 microns to 1 millimeter. So that's quite rational.
Sorry, and the hardcore is the minimal diameter of this vessel, so what is it?
Hardcore is effectively saying that there is a practically minimum space between the two branches of the tree, which may kind of compose this placenta. So it's effectively a growth constraint because it's an evolving structure. It cannot pack infinitely dense. So there will be physical constraint, which will, and of course there will be flow which pushes it apart. So there will be constraint which will create kind of hard repelling potential between the two points. So the smaller and another smaller bits. The fact that we don't allow them to collapse too much.
So there are some mechanism biophysical, physiological mechanisms which does not allow them to approach too much, right?
Yeah, well, I think there are lots of things. Some of it might be an imaging artifact as well, because we cannot resolve something beyond some point. But I think ultimately, uniformly random is very, so in some sense, as artificial as absolutely periodic, because it assumes that things can be infinitely close to each other. But if we think of porous medium composed of balls, then effectively it's saying that we don't allow all balls to merge, or at least not all balls to merge. And there might be several reasons why it has some features of for potential. One might be there's a flow in between, and the second is how the tree develops. Because what we're doing, we're just taking section of a porous medium composed of a extremely complex packed tree. But I think
Thanks.
I can answer why exactly this threshold is what it is. Does it answer your question or not?
Yes, thanks. Please continue.
And then we can think, all right, so we know a little bit more about different types of porous medium, different possible hypothetical types of statistics which can be described with. So we can think of, again, the simplest possible model for the transport in the disordered medium, in the porous medium. For example, if you think of a one-dimensional array, and this time that things correspond to these pores, to the tissue bits which extract solute from the passing maternal blood. So we lump all the features of the fetal vessels, all the feature of the fetal tissue, just in this point. So we neglect the special dimension, but instead we consider many of them, and we assume that they can be randomly placed according to one or the other distribution. And we have that two parameters, which tells the strength of the flow relative to diffusion and strength of uptake relative to the diffusion. So again, these two numbers. But the important feature is that we have a possibility to have a random uptake because of location of these things are unknown. And the question we want to answer is, first of all, what's the accuracy of the average concentration profile prediction by this model? And is this error is correlated over the domain or not? So we can say that, for example, each of these things can be weakly correlated or not correlated at all. But is it obvious that the global predicted concentration will have fluctuations uncorrelated at the global scale? So we want to do averaging of that system with these specific boundary conditions. So supplied some solute on the left and drain solute on the right, like a vein, and then prescribe a flow across it. Again, let's say a uniform flow. Again, it's idealization. And we can solve it exactly because that's a one-dimensional problem, linear problem.
Sorry. Could you please, why the concentration is zero, is assumed to be zero on the right side of the domain?
We can assume that the vein always large enough to extract all remaining solute. It's one of the possibilities. Indeed, there's other possibility to prescribe a flux, but it doesn't change fundamentally the conclusions. So, but indeed, it's just assumption and as simple as possible to us, because we will get a linear gradient, an average with this boundary condition. But we are free to choose other boundary condition, like prescribing fluxes instead of concentrations. But indeed, it's a good question.
Yeah, so, because here you assume a black sphere approximation, yeah, on the right. It is like a black sphere approximation, привержение черной сферы. Как будто у вас ток С на правой границе.
Yes, we just assume that it's an infinitely strong sink downstream. So assume that whatever left, it's always taken away, swapped away from the system.
Yeah.
But indeed, it's an assumption. So assume effectively infinitely strong flux at the end, which is an assumption. And we're now working with the PhD student who looks for a variety of boundary conditions and extending this in higher dimensions. But I think the message I want to tell would not depend so much on the specific choice of boundary condition, but rather on the statistics of the sinks in between. But it's a very good point. Boundary conditions do matter, but in a non-trivial way. So let's stick what we have. And the reason why I think it's nice, because it just shows that the average problem is very, very simple. So you just have a linear gradient between the inflow and outflow. So for all these three types of distribution, for uniform random, for hardcore, also known as mature type 2, and normal perturbation. So you have the sinks for a particular realization, blue circles at the bottom. So they all have the same average description. So if you think of only the averaging, like leading order, if you think of asymptotic description, they're all the same, absolutely not distinguishable. They're all ergodic, so to say. But what we're interested in is, first of all, does this average make sense if you compare it to the exact? And what about the correctors, the errors, if you compare to the averages? So the dashed line is the average, and the solid line is the concentration, the exact concentration around that average. And you see that staircase-like because the flow is relatively strong, so the concentration effectively is swapped between the sinks. There's not much diffusion happening. It's almost like a drift, kind of random wall. Okay, so if I take the difference between the exact, right, the solid and the dashed, we'll see that it's very different, surprisingly different between the normal perturbations and the uniform random or hardcore. So there, the solid line is the difference between the exact, which is this black, and the leading order, which is this blue line. And that's a random process, so it will vary to each point. And we also can predict the confidence or credible intervals. And it's totally different behavior when you have intrinsic regularity. Even if you cannot tell the difference immediately by eye, that is there, there, you can pick it up. Say that you expect low range correlations in the air at the scale of the entire array of the uniform random.
Excuse me, there is again a problem with the sound.
Shall I repeat the entire thing, or was it?
No, just the last 30 seconds.
Okay, thank you. So what I'm saying is that there is a fundamental difference between the uniformly random, Poisson, or hardcore statistics and the normal perturbations when it comes to predicting the features of transport. So even though by eye there's not much difference between the uniformly random and normal perturbation, but intrinsic structure makes a very big difference because your error in prediction between the leading order, kind of averaged, and the exact is much smaller and also uncorrelated effectively over the domain compared to the uniformly random or hardcore case. So that's quite surprising. And we can look into this further. So we can think of covariances, for example, how exact varies over the domain. And we can show, for example, that that's how it varies for the uniform random. So x is the distance. So that's the left. The inflow, that's the outflow. You can see that it varies. And
Sorry, the sound again is very bad, unfortunately.
Sorry, you again sound problem.
And now it's okay.
Okay. So what I'm saying,
I suggest that it turns on the long phrases somehow.
sorry about it. Well, please do drop me if something is missed, and I'll repeat.
Could you please return to the previous slide? I don't understand if we have uniform random distribution of points, then why random? Okay, yeah, I see. So okay, yeah. Please continue.
Excuse me, if I also can interrupt you to ask a question. So how the result depend on the density of these points, sinks?
It's a very, very
Is
good
it-
question. And that's what I'm going to answer in the next slide.
Sorry. By the way, once I have interrupted you, I suggested to discuss how we continue, because we are supposed more or less to finish, so we can spend, for example, several minutes more to come to some, I don't know, point in your presentation. So how do you estimate how we will do it?
So there are two parts. I think I need about 5 minutes to finish this part, maybe less.
Okay, so I suggest let's finish this part, and then for the second part we can do it the next time if you agree, right?
Okay, sure. Right.
Thanks.
So what I'm saying, I will try to be brief, but that's kind of the main point remains. So first of all, we see that even though there is very weak correlation between the points in the array in all of these cases, the error can be very strongly correlated over the length scale of the entire array. And the reason is because it's amplified by the transport equation, by the function of the sense. And we can predict this form of this dependence. But it remains the question, what's the magnitude? And how does it depend on the density indeed? How does it converge? And that's what we answer the question. So we talk about accuracy map. We have a variety of regimes dependent on the flow strength and uptake strength in the random array or normally perturbed array. And for example, if the reaction uptake is very strong, it doesn't make any sense to average anything because things vary dramatically from sink to sink. So there is no point of averaging. But even if it is possible to upscale, it will be very different, uniform random and normally perturbed. And the green region means that it's high accuracy and converges very quickly. And the yellow region means that it's low accuracy and converges slowly. And indeed, we can see that for relatively low number of sinks, let's say 10 sinks, that's slightly different regime. And here you can say that the red is the exact, sorry, the homogenized, the upscaled leading order behavior, average behavior, right? The same in both cases. But if you look at the specific realization, specific distribution of solute, you see that it's very far off of random realization. For the uniform random, and it always flows the normally perturbed, the same or the same sink density. What it tells you that if we think of, for example, what is the sink density of the center, what scale separation, neighborhood, and size. If I talk about periodic homogenization, then typically second order small, low, and it's linear scale separation. Still, it's very, very active. Periodic. And then sub jumps on top. Get 7 error. Some extent normally true. Having some undefined structure, if it's true, have structures randomly also better at average larger scale. Don't have any.
The sound is very bad again.
Okay, sorry. So, well, you heard hopefully that part. So I'm saying that there is a large loss of accuracy if you do not assume any intrinsic regularity. And the summary is that we can now have a framework for characterizing controlling the error, which will depend both on the physics, on the how strong is your flow, how strong is your reaction and uptake, or different solids will be different. And that will be the subject of second talk or part 2. But it's very surprising that we have long-range special correlation. And indeed, that will then be influenced further by the choice of boundary conditions. So we know that there will be correlations at the scale of the whole domain. And now the question, how does that is affected by the choice of boundary conditions? Indeed, it will. But at least we have a handle on how to combine the physics and the statistics to be able to know what is the average features and how much we can put our trust in any prediction. Thank you.
Thank you very much for this very interesting presentation, and indeed quite unexpected result that the organization is like more or less in all 3 cases you have a more or less a random realization of these sinks, but the result can be quite different. So it's really unexpected and very interesting. So please, if there are questions, so we can discuss them. Well, while our listeners are thinking about possible questions, I would like, well, to ask a question or to make a comment, which seems to me a little bit related to some of works we've done before. It's about not one-dimensional homogenization problem, but about two-dimensional homogenization problem. And I think it can be quite different. Really, so we did not consider exactly the same configuration. So we did not consider delta functions, but we considered small holes with some, and then the coefficient, I don't remember how it is called, the coefficient with the delta function in your equation, but the size of the hole, the value of this coefficient, and the distance between the holes. So depending on these parameters, the result of this homogenization can be quite different. So I really think that, and two-dimensional, and really you try in the two-dimensional problem and you try to pass to the limit, decreasing the size of the holes and the distance between the holes. So really there are several small parameters and the result can strongly depend on the relation between these parameters. And it's quite a difficult problem. But it's very interesting, and I think that it might be an interesting development of what you study in this work.
Thank you. If you can suggest what reference.
Well, I can send you what we did, but it's not, well, it's a parabolic equation because you consider it's a like elliptic equation or ordinary differential equation second order with this convective term. In our case, in fact, there are two different cases without and with convective term, but not stationary, but parabolic equation, time equation.
Yeah.
Okay.
Yeah, evolution equation, I see.
Yeah. And the question is how to pass to the limit when the size of this holes goes to 0.
Yeah, I totally agree. I think we didn't touch even non-steady problems. So all we did you can see on the bottom somewhat in a pilot where we're moving to the high dimensions, but we didn't touch on the non-steady flow and transport, which is of course very important too.
Okay, are there other questions or comments from our audience?
Вот у меня возник вопрос, когда вы в самом начале показывали на вот эти единицы обмена, где происходит обмен, самый мелкий, мне поразило, что структура, микроструктура, она тоже имеет в себе подструктуры разного размера. Грубо говоря, там были и сосуды и потолще, и потоньше. Вот где-то на одном из первых слайдов.
То есть, когда рассматривается это дерево, есть ли там какая-то фрактальность, да, в этом вопрос?
Нет, не совсем. В том, что размер, да, вот может быть даже следующий. Где-то вот там был, там, где был... Ну, вот, например, нет, я сейчас не найду, но, в общем, там были сосуды, в одном и том же рядышком стояли, и более толстые, более большого диаметра, и поменьше. То есть такое ощущение, что у вас есть сейчас параметр как бы расстояния между сосудами, да, есть параметр размера сосуда, но вот может быть вот этот размер сосуда это тоже некий параметр, который может быть распределен. Есть какое-то распределение в размере сосуда, и вот может быть это тоже может как-то влиять на какие-то свойства. Да, грубо говоря, вот у вас не просто там дырочки, вот как Виталий сейчас рассказывал, распределенные, да, и не просто вы их размер, в зависимости от их размера и расстояния между ними, но и распределение между ними, вот такое ощущение, что тоже может давать какой-то вклад в статистические свойства вот этой обменной функции.
Absolutely. Is anybody who doesn't speak Russian is present here?
Нет, похоже, что все.
Да, вы абсолютно правы. И одна из областей, в которой входят наши интересы, это попытаться понять, как можно соединить эти две среды, these two networks. Потому что there's such a thing called diffusion screening, я не знаю, как правильно перевести, когда будет, если вы, например, подаете высокую концентрацию с одной стороны, она может дойти до другой, может дойти в глубину этой структуры, потому что все, что модели, которые я показал, вы смотрите, вот point sync, это практически вся вот эта вся вот эта структура, она просто игнорирована, она вся конституционная.
Простите, у вас микрофон, по-моему, где-то сбоку остался, и поэтому опять пропал звук.
По крайней мере, я знаю, что он не является как дискриминационным к английскому, к любому языку. Что я говорил, то, что вы абсолютно правы, это называется скрининг диффузии, когда вы можете подать, с одной стороны, высокую концентрацию, например, вот на одном из этих объектов, если у меня видно курсор здесь, и если достаточно высокая скорость течения в этой части, то может до другого элемента не дойти концентрация, то есть все будет поглощено с одной стороны, то есть может быть несимметричный транспорт.
То, что называется внутренняя диффузионная область. Насколько...
Да.
Да.
То есть мы это все на данный момент игнорируем и считаем просто как точечным стоком с эффективной реакцией.
А скажите, пожалуйста, вот я, наверное, в самом начале немного упустил вообще про структуру. Вот на этом слайде видно, вот с правой стороны есть вот эти кружочки такие неправильной формы, замкнутые кривые, а внутри зелененькие точки. Значит, капилляры – это вот эти зелененькие штучки.
Да.
А вот эти вот коричневые линии вокруг – это что такое тогда получается?
Это ткань. Это, поскольку это не просто сосуды, которые буквально окутаны кровью. Если вы посмотрите вот на следующий слайд, то вот видно, что это происходит. Это вот как бы этот капилляр, между ним есть тонкая прослойка, а снаружи уже материнское кровообращение. И это самая тонкая часть. Но есть, конечно, часть, которая смотрит в другую сторону, которая просто будет разная соединительная ткань и просто ткань. То есть это как бы это дерево в дереве. То есть здесь как бы три области. Есть дерево, вот сосудистое дерево ребенка, плода. Есть область материнская, пористая среда, и есть как бы ткань, которая окутывает это дерево. То есть это дерево сидит в дереве, оно то есть, как минимум 3 ряда.
А вот эта вот промежуточная ткань, то есть она не принадлежит ни тому, ни другому, да, это просто некая промежуточная среда, через которую происходит обмен вот всеми этими веществами. Да?
Но и капилляры, и ткань это это принадлежит ребенку, то есть плоду, это его развивается от плода. И это то, что вы видите, это как бы просто в принципе как есть цитата Бернарда Шоу, что мы никогда не ведем себя как деревья, за исключением первых 9 месяцев. И это является просто неотъемлемой частью кровообращения плода. То есть это просто его продолжение.
А скажите, вот это дерево, нарисованное слева из кровеносных сосудов, его структура, я полагаю, изучалась, да? То есть, допустим, вот у взрослого человека вот это большой круг кровообращения или легочное кровообращение, структуры этих сосудов, их взаимосвязь, их размеров у разных поколений более или менее определена, конечно, хотя она, конечно, очень сложная. Анастасия, здесь присутствующая, как раз этим занимается. Я думаю, она может прокомментировать. Здесь, в плаценте, это изучено или нет?
Плацента намного менее изучена, чем многие другие органы, и этим это интересно, потому что она долго считалась таким временным, не особенно интересным органом, который просто как-то так появляется и исчезает. Пока они стали понимать, что даже если ребенок родился здоровым, то там каждый, например, грамм или там 10 грамм, который он не добрал, то он определяет его практически полностью, программирует его здоровье и интеллектуальный потенциал. То есть сейчас намного просто, во-первых, не было данных о плаценте экспериментальных, поэтому модели все были очень эфемерные. А во-вторых, действительно не было такого интереса, и трудно было найти поддержку для проектов. Поэтому сейчас все интенсивно группы изучают в Англии, в Новой Зеландии, и в других странах. Но я могу сказать, что это намного менее изучено, чем другие органы.
То есть структура этого дерева пока еще неизвестна, да? То есть, допустим, делятся сосуды на 2 части или на 3 части, и соотношение между диаметрами и длинами вот этих отрезочков неизвестны пока.
Частично известно, но проблема была именно в том, чтобы соединить все масштабы. Известно на очень больших, для самых крупных сосудов известно, то есть люди всегда смотрят на плаценту, видны эти сосуды, они прям видны невооруженным глазом. Люди, часто биологи делают фотографии, пытаются предсказать, как глобальная сеть влияет на здоровье плаценты. Но когда это уже доходит до маленьких, нету континуума от больших до маленьких сосудов.
Да. Анастасия, вы, если присутствуете, напомните, в легочном кровообращении там сколько? 14 поколений, да, сосудов?
По одной модели выделяют 15 артериальных, 17 венозных, а по другой по 15. Но там на каких-то уровнях они начинают, это уже статистическое приближение в какой-то мере. Но эти модели, они делались, я так понимаю, заливалась реальная легкая человека, там, вытравливалась, получали они какую-то полимерную модель сосудов, и дальше по ней считали как-то эти сосуды с учетом того, что не всюду мог этот эмболизат пройти. Но то, что посчитали, 15 уровней выделяется. Да.
Здесь точно такая же проблема, точно так же, то, что вы видите слева, получено с помощью специальных контрастных резин, рентгеновских контрастных, которые тоже имеют такие же ограничения, что, во-первых, они очень высокую имеют вязкость и сильно деформируют сосуды. И я подозреваю, что они далеко не проникают в совсем-совсем маленькие части.
То, что построено для легочного, там говорится, что это для артериол и венул, а для капилляров уже не проникает в капилляры.
Но сейчас с возникновением того, что я показал в самом начале, эта модель, она, в принципе, почти воспроизвела то, что было получено с помощью традиционных методов, потому что на синхротроне можно получать информацию сразу обо всем, не вытравливая. То есть это получено с цельного куска ткани, где можно залить вещество контрастное, но при этом не растворяя ничего, получить одновременно. И это буквально данные, которые еще до конца не напечатаны, вы видите там ссылку внизу. Но это, многие считают, что это будущее направление с точки зрения данных геометрических. И, возможно, насколько я знаю, это используется во всех других моделях тоже. Не только для плаценты, и мозг изучают так, и лимфатические узлы. И уверен, что, если нужно, то и легкие тоже. На маленьких животных, конечно, не на человеке.
Спасибо. Есть ли еще вопросы у наших слушателей? Если нет, то давайте...
Да, у меня,
Алексей, давай.
Один вопрос, да. Вы сказали очень интересную фразу, да, что каждый 10 грамм, да, влияет на развитие ребенка, да, то есть получается, что одна из важных проблем – это изучение проницаемости плаценты, да, для нутриентов всяких, да, для, и так далее, и всего. А еще какие здесь есть, так сказать, не мейнстримы, но проблемы, такие, ради чего стоит, первое я понял, да, проницаемость влияет на, рост ребенка на его на вещество, которое проникает. А еще, вот, например, гемостаз в плаценте изучается или нет? Или, скажем, иммунный ответ какие-то. Вот, да, гемостаз. Вы не слышали вот насчет тромбозов в плаценте, эмболии, может быть, вот там не знаю.
Все очень хорошие вопросы вы задаете, но на них нет ответов, насколько мне известно. Биологи давно изучают плаценту как с биологической точки зрения. Они смотрят на иммунную составляющую, на воспаление в плаценте, но с точки зрения математической модели или механики, даже вот с самыми простыми вопросами, как транспорт кислорода, это далеко-далеко еще даже не приближается к средней стадии развития модели. И с точки зрения реологии, я даже об этом говорил, это на самом деле больше вторая часть, которую, спросите меня еще раз об этом в следующий раз. Совершенно нет даже фундаментальных моделей, как описать течение крови в пористых средах. Вообще, в принципе, нету фундаментальных принципов. То есть даже в сосудах это сложно, но есть модели, которые описывают реологию сосудов. В пористых средах моделей нет вообще.
Это интересно, кстати, очень интересный вопрос, поскольку мы очень много занимаемся как раз-таки течениями крови в кровеносных сосудах и свертываемостью. То вот такой вот неожиданный поворот течения крови в пористой среде, как-то я не знаю, но лично я с этим не сталкивался, с этим вопросом. Вот, действительно, очень интересно. Хорошо, пожалуйста, если... Спасибо. Если других вопросов... Вопрос?
Ваши вопросы.
Да. Хорошо, тогда, если других вопросов нет, то мы еще раз вас благодарим за очень интересный доклад. И я бы хотел попросить Анастасию по поводу, когда мы можем провести продолжение, второе заседание этого семинара.
Будет ли удобно, сейчас скажу, 6 мая, через месяц, побольше.
На данный момент я могу, да.
Да, конечно, тогда давайте назначим на 6 мая. Сейчас, минутку, я на всякий случай. Но в любом случае у меня четверг зарезервирован для семинара, так что я думаю, на всякий случай я проверю. Да, все в порядке, так что давайте тогда на 6 мая мы запланируем тогда продолжение вашего доклада. Я надеюсь, что как-то то, что мы разбили на две части, это не вызвало у вас большого возражения. Вот. Но действительно, мне кажется, что если есть возможность спокойно обсуждать и разбираться в вопросах, то это удобно и интереснее слушать. Спасибо еще раз, спасибо всем присутствующим, и тогда до новых встреч на нашем семинаре. До свидания.
До свидания. До свидания.
До свидания.
До свидания.
Спасибо.
До свидания.